Dear students, today we are talking about the antibiotic spectrum, and in the broad sense we can say that antimicrobial spectrum. By the antimicrobial spectrum of an antibiotic, it means that the range of the microbes or the number of the microbes or the different type of the microbes an antibiotic or antimicrobial drug can kill. Based on their spectrum, this it can be classified as like this: narrow spectrum antibiotics, broad spectrum antibiotics, and extended spectrum antibiotics. Narrow spectrum antibiotics. Narrow spectrum antibiotics means it can kill a narrow group of the microbes. For example, the aminoglycoside, sulfonamides, only effective against the aerobic organisms. Therefore, you can see that the aminoglycosides is used to give in the treatment of tuberculosis, because Mycobacterium tuberculosis is an aerobic microorganism. Nitroimidazoles only effective for anaerobes. Glycopeptides, bacitracin, it is active against only gram positive bacteria. So in the narrow spectrum antibiotic, we see the spectrum of the killing of the microbes is very narrow. Coming to the broad spectrum antibiotics. Broad spectrum antibiotics against both gram positive as well as the gram negative organism. For example, fluoroquinolines, tetracyclines, and we can also talk about the third and fourth generation cephalosporin. They are the broad spectrum antibiotics. Extended spectrum antibiotics, as it is given, they are active against the both gram positive as well as the gram negative bacteria. In addition to this, they are also effective against resistant bacteria, and the examples under this category are quinolines, ampicillin, chloramphenicol, amoxicillin. So this is all about the spectrum of antibiotics. What are antibiotics? Antibiotics are those which are obtained from different microorganisms and are used to kill the different microorganisms in a very low concentration. Now coming to the standardization of antibiotics. What do you mean by the standardization of antibiotic? As you see in this slide, the standardization of antibiotic refers to the assay of individual antibiotics and their preparation to evaluate the potency of antibiotic, that is the actual and the apparent measured effect is the degree of the inhibition of the growth of a suitable strain of microorganism. It means that you are checking the potential or the killing power of an individual antibiotic with reference to a standard antibiotic. So the assay of the antibiotics can be categorized can be done by two methods. We have cylinder plate methods, we have turbidity method. Now we have methods used for standardization of antibiotics. This is most important, and sure you will surely get this type of the question in the examination, because this is very important. And there are two methods. First is the cylinder plate method. In the cylinder plate method, it is based on the measurement of the zone of microbial growth inhibition immediately surrounding the cylinder, containing various dilution of substances under test that are placed on the surface of a solid agar medium already inoculated with the culture of an appropriate microorganism. What happen in this? We will prepare the Petri plates of agar medium, and then we will inoculate within the microorganism. After inoculation, they are incubated at a definite temperature and time. After that, the test sample is placed on these agar plates, and finally what we will measure the zone of microbial growth inhibition. ठीक है? You can see here the zone of inhibition produced by the test compound is compared with that produced by the known concentration of the reference standard and compound. Now finally we will compare the zone of inhibition of the test compound and with reference to standard compound. You can see these are the Petri plates, and zone of inhibition comes in the circular form. Okay, so this is the cylinder plate method. Now coming to the turbidity plate method. Its principle is based on the inhibition of inhibition of microbial growth as depicted by the corresponding measurement of the turbidity, and it is the transmittance of a suspension of the microorganism in a liquid medium into which the graded amount of the test samples have been fully added. You can say that यू कैन से दैट कि इसमें हम क्या करते हैं ना, टर्बिडिटी को मेज़र करते हैं. टर्बिडिटी मेथड्स होते हैं, इसमें हम सस्पेंशन बनाते हैं माइक्रो ऑर्गेनिज्म एंड देन वी विल चेक देयर टर्बिडिटी. एज ए रिजल्ट द चेंजेस इन द ट्रांसमिटेंस कॉज्ड बाय द टेस्ट कंपाउंड कंपेयर्ड विद दैट, यानी जो टर्बिडिटी टेस्ट कंपाउंड देता है और जो टर्बिडिटी आपका स्टैंडर्ड कंपाउंड देता है उसको हम कंपेयर करते हैं, और इसको हम टर्बिडिटी मेथड बोलते हैं. तो हम कह सकते हैं स्टैंडर्डाइजेशन ऑफ एंटीबायोटिक्स हम दो मेथड के द्वारा करते हैं. एक सिलेंड्रिकल प्लेट मेथड होता है, दूसरा टर्बिडिमेट्रिक मेथड होता है. बहुत सिंपल से, बहुत इंपॉर्टेंट टॉपिक है, एग्जामिनेशन में बिल्कुल आ सकता है. इसको अच्छे से तैयारी करें. थैंक यू.